All right, hello to my friends who are joining us via recording. Today is April 30th. We are reviewing for our lecture final exam. Remember, the lecture final exam opens up on Monday. Let me get the date for you, Monday, May 4th. So this should help us out. We're primarily going to focus on units 1 and 2 for today's session. Might move on to unit 3. We will also be reviewing for that exam tomorrow as well. So I'm going to pop over to my chat and see what we were thinking. We want to know about the different types of channel proteins. So maybe we can talk about transport across the membrane in general. Okay, so we want to review the learning objective about the charges on a neuron, and that one ties in really well to what we did with Unit 3 stuff. So the way I structured the course for you guys is we talked about it first way back in Unit 1 when I was like blowing your mind, right, with neuron stuff. You're like, what in the world? And then we talked about it again in Unit 3. So we can definitely talk about neuron membrane charge. And what I'll say too is if there's stuff in Unit 1 that is a little dicey for you that relates to that, if you got it in Unit 3, you're probably set. But we'll definitely talk about that. Okay, so I've got transport proteins on my list. I've got neuron membrane charge stuff. What other topics do we think we want to cover today? Are there other things that we, okay, so the ions and the stuff that's inside the cell, for sure. Ions, molecules in cell. Let me just ask for my own reference so nobody else will see this. If you have had a chance to start reviewing for the exam, can you raise your hand for me? If you haven't started reviewing, that's totally fine. I'm just curious how many of you have started to review. It's okay if you haven't, like, it's still early, right? Okay, so several of us are still waiting to start. Let me see really fast. The one thing I didn't bring into the room with us today is that list of learning objectives. So let me convert it really fast to a PDF. What we'll do is we'll start by looking at that list together, just as a heads up for us. These objectives that people are mentioning come from this list that I'm going to import here for us. So in, I apologize, there we go. I believe it's in the main lecture resources folder. You have a document that says final exam learning objectives. That's what earlier today when Eileen mentioned something that pregnancy brain is now stealing from me. Oh, asked how many questions we had on the exam. I told you to look over that list and kind of estimate. That's what I was talking about. So final exam learning objectives, let me pull it up for you. Here's the document that you have. Get at page 1. And zoom in a little bit. I gave it to you guys in Word format so that if you wanted to go in and type up your notes underneath each of these learning objectives, I wanted to give you that option. So you can download it. If you'd rather type instead of handwriting, you're welcome to. It's not a PDF. But this is a list of the topics that we've covered all semester, the ones that I want you to be reviewing for the exam. So the first unit that we did together was the first 4 lectures. For my Friday friends, it was about the first 3.5 lectures. So yours is maybe a little bit different toward the end, but the material is all the same. So just to give you kind of an overview of what did we even do in this class is basically what this list of learning objectives does. So for example, the first learning objective is always going to have those list of terms on it. Remember from lecture exams with me in class that I don't ask you definition questions. What I do ask, or what I do assume, is that you know what all of these words mean, because I'm going to use them in my questions or use them in my answers, assuming that you know what they mean. So don't spend forever going through and defining these words. I promise you there won't be a definition matching, but these are those words that I want to make sure that you understand. Then we get into the content learning objectives. So for example, the first lecture that we did together was about anatomy and physiology, how those two branches of science are related to each other. So let me ask the question that I always ask you guys in class. Anatomy and physiology, related or not related to each other? Are these branches of science related? Yes, they are. They're related, or the word that we used way back in Unit 1 is that they dictate each other. So if I know the anatomy of something, I can predict the physiology. If I know what something does, the physiology of it, I can predict what structures I need. So this first learning objective just asks you, go back and review our first lecture together, see how we made some of those predictions. On the exam, I may ask you to predict the anatomy or predict the physiology of something based on whichever one I give you. Somebody mentioned in the chat maybe that we could talk about brown fat and white fat. We could certainly talk a little bit about that. There's a note on here about the organelles, for example, and I referred you guys to the lab packet because remember when we talked about the cell, we didn't talk a lot in our lecture notes about what the organelles do, but you can expect at least one organelle question on our exam. So this is a list of the topics here. So all the way through, I think this is everything, yeah, for Unit 1. So there are 18 learning objectives for Unit 1. Expect to have about 18 questions, maybe a little bit less than that from Unit 1 material. Then we move on to Unit number 2. Again, we've got a list of the terms that I'm going to assume you know what they mean. If you're reviewing the material here in the other learning objectives, you will be bumping into these words anyway, so use them or don't use them, it's there for you. Things like those levels of protein structure, make sure we know what happens at each of those levels, make sure we know how those different levels form. If we have time today, this would probably be a good one for us to review just because it's good basic knowledge to have about proteins. This was also the unit, if you remember, we talked about Creutzfeldt-Jakob's disease, the mad cow disease, essentially, the human form. So make sure to review a little bit about those, about transcription and translation, and then we transitioned into talking about our first organ systems. So that was the skin, that was the skeletal system. So I'm not going to go through and look at all of the learning objectives with you. And I know a lot of us are just starting our review process. So maybe plan to bring tomorrow or especially to Monday, plan to bring with you, excuse me, some more specific learning objectives that you want to review. We'll review some of these topics here, but again, use this as your outline. I know at one point in the chat, Eileen said it's a little bit intimidating. I would say try to reframe it being intimidating, to reframe it to being, wow, I've learned a lot this semester. Let's make sure I go back and see all the things that I've learned. Because when we first started this semester, you probably didn't know what half these words meant. Now you at least know what they mean. Whether or not we can, go through right now and explain how I form the structures, maybe not yet. But we at least have some working knowledge of all this stuff. So don't let it be as much intimidating as here's a reminder of everything we've done this semester. I know this stuff, I've got this stuff. So please check out that final exam learning objectives list for you. I promise if you go through and study all of this material, you will be prepared for that exam. Okay, so let me bounce back to my chat and make a list of the other things that we mentioned. Ions, okay, types of bonds. We can definitely do that. Okay, and then we can do a little bit with Central Dogma stuff. I bet that'll fill our time today. And with brown fat, white fat, I'll make sure to hit that. Okay, so my goal is to hit the topics that you guys mentioned for me. If we miss something that you really want to talk about, feel free to bring it back tomorrow. And we have one final review session on Monday as well. So let me go back to all the pictures that I pulled out of unit number 1. Yeah, so the question in the chat is the final exam still replacing our lowest test grade? Yes, it is. So if you do better on the final exam than on another lecture exam, that will replace that grade. If for some reason the final exam does not go well for you, I'm not going to penalize you and replace a higher score with it. But the final exam does always count in your grade. It's worth 10% of your course grades. So please study for it. Study for it just like you study for those normal exams, and see it as an opportunity to raise your grade. But again, I'm not going to like double punish you for not doing well on it. I think you guys are going to do okay on it. I think usually the final exam goes well for people because people know how to prepare. Okay, first topic we hit in pictures. Zoom out. So sorry guys, I swear like my voice is fine until I sit down on the camera, then it gets funky. One of the learning objectives asks you guys about the ions and the molecules that I find inside and outside the cell. This probably graph, help me out, is this lecture number 2 or number 3? I don't know if anyone has their lecture notes in front. Okay, so this is a graph from lecture number 2. Awesome. Okay, so let's start here with lecture number 2. Okay, Rosa's saying it's on page 3. Awesome. Okay, so way back in the day, when we first looked at this graph, we didn't fully understand the importance of this graph. Hopefully now we understand the importance a little bit more, now that we've started talking about more of these things. But this is the graph that we use to talk about what's going on inside and outside the cells. So inside and outside the cells that we see in our body. I guess I didn't bring over my, my legend here for us, so let me type it in here. Blue, help me out in the chat. Was blue interstitial fluid? Am I remembering that right? Okay, interstitial fluid. Interstitial fluid what was red? Yeah, I knew that yellow was, okay, I thought red might be plasma. Perfect, okay. And then we'll get our, I'm gonna make it orange just because yellow doesn't show up very well. So I'll type yellow. Yellow is intracellular. Okay. When we first looked at this graph, the goal of this graph here was to tell us what I have a lot of and what I have a little of in the different fluids of the body. This is going to matter because when we start talking about things like the way that neurons send messages, these things like sodium and potassium should ring some alarm bells for you. I always ask you when we're talking about cells, right? I always ask you about our favorite, the salty banana. So the salty banana came from this graph here, way back in the day. So let's draw ourselves a salty banana. My salty banana cell here, it looks like a sickled cell, right? It is a banana, and we're looking at our graph here too, so our graph can help us out. Inside a normal banana, we find a lot of one ion, and I can see it on my graph here. There's a really big yellow bar for this ion. What ion do I have a bunch of inside the cell? Yeah, a whole lot of that potassium. I'll put its whole name and then I'll abbreviate. A whole lot of potassium inside this cell, just like we'd have inside a banana, potassium. Now, we said that the bananas that are our cells, we said that these are kind of like supercharged modified bananas because the other thing you see a lot of on the graph that we can see here is we also have a lot of proteins inside our cells. There's one other molecule that we always like to add to our list of this salty banana. Yeah, the other thing we always like to add is a whole lot of glucose. Now, your learning objective asks you to explain the importance of these various things that are inside and outside cells. Who can help me out? Why is glucose important? What makes glucose important? Yeah, glucose is something that my cell can use to build energy, right? So this is my energy source, or the way I sometimes talk about it is it's my cell's favorite food. Favorite food. It makes sense that my cell is going to want to have a lot of its favorite food inside of it to make sure that it stays nourished, to make sure that it has what it needs. When we talk about potassium, potassium is something that I use at the same time as a couple of my other ions that we talk about. Potassium is something that I use to help me generate a normal charge on my membrane. Who can remember my technical name for the normal charge that a cell has? The inside's not the same as the outside. What's it called that I have that normal charge? We got a couple ideas. Yeah, so the cell is polarized, right? The name of my normal charge on a cell when it's polarized, it has a big long name. Resting membrane potential. Remember that word? Resting membrane potential. You can abbreviate it RMP, resting membrane potential. When we talk about the importance of potassium, what makes potassium important is that it helps to generate the resting membrane potential on my cells, the normal charge that they have. Different concentrations of potassium inside compared to some of the ions on the outside help us with that. Proteins do a whole lot of different things inside my cell. Can we brainstorm a list or use our packet to help us out there? What are the many different things that proteins inside a cell do for us? What are some of those jobs of proteins in a cell? Yeah, first job of the proteins is to give that cell its shape. Absolutely. So the technical word, if we want to use an oral word for what proteins that give me the shape do, it's called the cytoskeleton. Cytoskeleton, that's the shape of my cell. So one of the functions of proteins is they give the cell its shape. They make the cell's cytoskeleton. Another function that they do that someone mentioned in the chat for me is that proteins act like enzymes. Proteins act like enzymes. So enzymes are specific proteins that can do a lot of different functions in the cell. We talked about a lot of different enzymes this semester. Here, I'll type this off to the side for us. One of the enzymes that we talked about was called RNA polymerase. RNA polymerase. Does anyone happen to remember what RNA polymerase does in a cell? Can we use its name to figure out what RNA polymerase does? Yeah, so it's kind of making a copy for me. Yeah, it's making an RNA copy of my DNA information. So hopefully today we'll have a little bit of time to talk about the process of transcription, where we make specifically mRNA from DNA. That's the job of the enzyme RNA polymerase. It makes RNA from DNA. So that's one example of an enzyme in the cell. Your lecture packet also talks about an enzyme that's called lipase. Lipase meaning it breaks down lipids, like its name suggests. Hey, let's see if we remember this one. This one was from the muscles chapter. Atpase. What does Atpase do? Can anyone guess? Yeah, it breaks down my energy, right? It breaks down ATP. So breaks ATP. When we were talking about our types of muscle tissue, I told you that not every ATPase enzyme works at the same rate. So the ATPase enzymes work at different rates. Some are fast, some are slow, which means that some of my muscles contract stronger, some of them contract weaker. So all of these are examples of enzymes, proteins acting as enzymes. The last thing that proteins do, and a couple of us mentioned this as well, is they help us to do, I'm going to put in quotes, the overall function of the cell. So the example that I gave you guys with cells helping us with, or with proteins helping us with the overall function, that was when we talked about hemoglobin, right? So the job of the hemoglobin protein is to help me transport oxygen. I fill up my cells with the hemoglobin protein, and having that hemoglobin protein allows that cell to do its job, allows it to send its information. So here are the three main components that are inside a cell. Potassium, which is involved in polarizing the membrane, giving us this resting membrane potential. Glucose, that was my primary energy source, and then those proteins that help me out with all kinds of things. Remember, we said proteins are what I have the most of in the cell when it comes to those macromolecules, because they're doing all kinds of different things inside the cell. Outside the cell, there's two main things that we talked about. So I'm just going to put these things in blue. What are the two things on my graph that we see over here that I've got a whole bunch of outside the cell? What's there a bunch of on the outside of my cell, my salty banana? Yep, this is where it gets salty, right? So sodium is the first half of salt. Sodium, that's Na+. And we also have chloride. Oops, chloride. That's Cl-. When we talk about the function of sodium outside the cell, the biggest function of sodium is the same thing as potassium. So sodium is really involved in that resting membrane potential, making the cell polarized so that it's more negative on the outside compared to on the inside. But the other thing that we should mention with each of these ions, whether it's sodium, chloride, or potassium, is that it's involved in neuron signaling. Neuron signaling. When we're talking about the way that neurons work, what's the specific job of sodium in helping a neuron work? Can anyone remember what sodium does when a neuron's trying to do its job? What does sodium do? Yeah, so we're getting that technical word in there. Technically speaking, sodium is what depolarizes the membrane. Depolarizes the membrane. So I have to have sodium to be able to do that, and I have to have sodium that's really highly concentrated outside. Because remember, sodium rushes inside to depolarize the membrane. So when we're thinking about the importance of sodium, two big things. It helps us with this resting membrane potential, but it also helps me with depolarizing the membrane when we're talking about neurons, when we're talking about muscle cells. With chloride, again, it's kind of the same deal. I guess I'm going to put it underneath so we don't bump into things. Chloride is involved in neuron signaling as well. Chloride is the ion that I can bring in when I'm getting an inhibitory message on a neuron. So when I get an inhibitory message, what it does to my membrane is it's called hyperpolarizes the membrane. Still going to bump into words here. Hyperpolarizes the membrane, meaning it makes the membrane extra negative. So one of the big things that chloride is important for is for helping me when my neuron is getting a message. Remember that I can open up a channel for chloride, which lets the chloride rush inside and makes the membrane more negative. So when we're considering the function of these various things or why they're important, a lot of them we're gonna think about and filter through the lens of neurons. So sodium depolarizes my neurons, chloride would hyperpolarize my neurons. Potassium technically does the same thing, right? Potassium leaves my cell and makes it more negative too. So one of the learning objectives that you guys are using to prepare for the exam, first half of it is knowing where stuff lives. Does it live on the outside? Does it live on the inside? That's your salty banana. So always remember that salty banana. And then the other half of it is knowing what those specific things do. And that's what we kind of outlined here. A lot of it, again, think about it with neurons. I just about tried to write the word neurons. That would have been a huge mistake. Neurons. Think about what these things do with neurons, because that's the big type of cell that they play a big role in. Questions or thumbs up? What are our thoughts? Yeah, Eileen says it's easier when I think about it with neurons. Do it. Absolutely. When we first talked about these things, we didn't know neurons yet, right? But now we know them. So absolutely use those neurons to help you out to think about it. Ah, peace sign. I like it. While we're at it, let me go down and see if I can find. Let's go down to my favorite graph, right? Because it relates to these ions and stuff. Can you guys believe that way back in Unit 1, like the very first unit together, I made you guys learn this? Like, can you believe I was that teacher? So one of the questions that you guys had was about the membrane charge and about how the charge on the membrane changes. I mentioned it before and I'll say it again. This is 100%. Yeah, Deepa's like, it makes sense now. Yeah. This is 100% exactly the same as Unit 3. This graph has not changed. This was our application of the cell concentration stuff. So you guys already told me this, so let's add some notes to our graph here. Number 3 right here is when the process of depolarization is occurring. Yeah, and so like Eileen mentioned in the chat, and you guys told me before, depolarization, when the membrane charge gets positive on a neuron, that's thanks to sodium. Oops. Is sodium coming in or is sodium going out? What happens here? When I depolarize, sodium, yep, comes in. Sodium enters. Sodium enters. Over here on part 4 of my graph, this is the process called repolarization, where I'm trying to get my neuron back to normal. Yeah, and repolarization involves one of those other ions that we just said was important on our graph. The ion it involves is potassium. Potassium comes in or goes out. What happens when we repolarize? Yeah, potassium leaves. Potassium leaves. Hey, way back in the day, it was really terrifying for us to try to figure out, is stuff coming in or is it leaving? But remember, it's that salty banana, right? It's that salty banana again. If I open up a channel for these guys, they only go the way that they normally want to go. So sodium only has one option. It can only come in because there's so much outside. Potassium only has one option. It can only go out because there's so much inside. So depolarization, when my charge goes up. Repolarization, when my charge goes down. What's happening at these various places is when we have some of those types of ion channels that we talked about in the semester. So the types of ion channels that we talked about. We talked about, let me list the ones here we're going to mention on our graph. Voltage-gated ion channels. We talked about chemically gated ion channels. Way back when we first talked about this graph, it terrified us because we just learned what these two types of graphs or these two types of channels were, right? We were trying to learn what these channels were, and then we were trying to put them on a graph, and it was so overwhelming. But now we got this stuff down. So let's do some definitions here. Voltage-gated ion channels. Here, let's ask my favorite question because it's been a while. A gated channel is or is not always open. A gated channel is or is not always open. Absolutely right. Gated channels are not always open. Okay, so when I talk about a voltage gated channel, opening up the gate, Eileen mentioned this for us here, opening up a voltage gated channel, it's all about charge. Charge opens gate. Charge opens gate. When we talked about chemically gated channels, what's going to open up a chemically gated channel? Not a trick question. Yeah, just like its name says, right? It's all about, so I'll put in caps, chemicals open gate. Chemicals open the gate. Now, when we're looking at this particular graph, the type of, because we're looking at the graph, remember, on a neuron. This is a neuron's membrane charge. When we're talking about chemicals opening gates on a neuron, there's one specific type of chemical that opens up gates. What's the specific type of chemical that opens up gates on neurons? What kind of chemicals open up these gates? It's a big long word, right? I'm making you type a big long word in the chat. Yeah, or we can just abbreviate, right? Yeah, so the big long word for it is neurotransmitters. Neurotransmitters, abbreviation NT, right? When we are talking about chemically gated ion channels, the specific type of chemicals that open these channels up are the neurotransmitters. When we talk about voltage gated channels, that's always, as we know now inside and out, that always has to do with charge. Okay, so on my graph here, the place where we switch between these two types of channels is a place called threshold. Remember, from Unit 3, threshold is really important because at threshold, my charge has changed enough to open up which of these kind of channels? Here, I'll let you abbreviate. Is it going to be VG or CG that opens up at threshold? Who opens up at threshold? Yeah, so those voltage ones, right? Voltage gated. Voltage gated. Would help if I could spell. There we go. Voltage gated. At this point on my graph, threshold value we talked about, this is when, in particular, my voltage-gated sodium channels open up. My membrane charge has changed enough that I know I need to send a message to my neighbor. Remember, I did the process of summation, and I changed my membrane charge, so I open up the voltage-gated channels. That allows me to bring in all kinds of sodium. We get all the way up here to positive 30. Suddenly I'm super positive. My membrane charge is wrong. So which kind of potassium channels are opening up here and are helping me down here? Is it chemically gated or voltage gated that I have on this side? Yeah, a couple of us are chiming in. Again, on this side, it's those voltage gated ones again. Voltage gated. The charge is wrong. So I open up my gate. Potassium is going to spit out. My voltage gated channels will start to close as my charge gets closer to normal. But remember, it doesn't close fast enough. We get a little bit too depolarized. We get hyperpolarized, to use that word from before. So the other kind of channel on the graph that we talked about way back in Unit 1 was those chemically gated channels. Remember, the chemically gated channels are here at the very beginning. So before threshold, it's those chemically gated channels. And remember, in Unit 1, we didn't talk about how there was different kinds of chemically gated channels, how some of them made a charge go up, some of them brought the charge down. You know that from Unit 3 now. But the big thing to know in the context of Unit 1 is that here's where on my graph there's chemically gated channels. Here's where my graph I've got voltage gated channels. And there's one other type of channel protein that I use on this graph. It's the one that I use over in this part to get me back to normal. What's the other kind of channel that I use on this graph over here? So Ariel mentioned leakage. Yeah, so I was going to say just leakage. When we talk about the other type of channel that's on this graph, they're called leakage channels. Leakage. I got some caps lock on. There we go. Leakage channels. Leakage channels, this would be an important idea for us here. Whenever I talk about a leakage channel, there is no gate. One of your learning objectives was comparing gated channels to leakage channels. Leakage channels have no gate. So we have no gate. Does that mean they are or are not always open. Are or are not always open. Yeah, I mentioned they're constantly leaking. They're always open, always open. They're constantly leaking. Think about them dripping. I gave you guys the example of a leaking faucet. So that's what a leakage channel is. It's a channel that's always open. It moves things really slowly. It's always going to move them from high to low. So a leakage channel, always open. Gated channels, not always open. An important difference between them. Let me see if I have a quick picture. So here I'll toss up, this is earlier in that packet, just to show you a picture of, we'll go there, here we go, okay. Show you a picture of a leakage channel, okay. A leakage channel, there's always this passageway that molecules can get through. Molecules are always going to use leakage channels. They're constantly going through and undoing the hard work of polarizing the membrane. That's what a leakage channel does. It's just kind of like I said, when you have a leaky fit, you're always dripping little pennies, if you will. We're losing water or losing money out of a leaky sink. So constantly things are dripping through these leakage channels, coming through slowly. So Eileen says it's always open. Absolutely. So here's some notes about our leakage channels. They're always open. Always open. And they're going to, I'm going to put drip in parentheses, meaning things move slowly. Things move slowly. So leakage channels, as I see right here, always open, constantly dripping things slowly. Let me ask you this question, because this has been a while. When we talk about leakage channels, are they doing active transport or passive transport? It's been a while since we talked about those words. Active transport or passive transport? Yeah, a few of us have chimed in. When I talk about leakage channels do passive transport. Passive transport. There's a few things that passive transport means. Passive transport, what does that mean about energy? What does passive transport mean? I do or don't need energy to do passive transport. Yeah, I don't need energy, so no energy, no energy. And the way that I can move things that doesn't take energy, I saw this pop up in my chat too, I have to move things from where there's a high concentration to where there's a low concentration. So these are the dripping faucet. They're always going to move things slowly, but the caveats with them is that I can only move things from high concentration to low concentration. So if we're thinking about that salty banana cell, here's my little cell, right? We can only ever move things the direction where they're going from a high concentration to a low concentration. So let's remind ourselves, this is going to look really big. What's the ion that I have a bunch of inside the cell? I'm just going to ask you this forever, every day, all the time. Yep, potassium. Whole bunch of potassium inside. Whole bunch of potassium inside. And I got two ions on the outside. My salt. My salt on the outside is made of sodium. Yep, my sodium positive. My chloride negative. When we talk leakage channels, they can only leak the opposite directions. So I can only leak out potassium. I can only leak in sodium. I can only leak in chloride. Leakage channels, they always leak from high concentration to low concentration. So leakage channels do passive transport. No energy required. We go from high concentration to low concentration. We are always open, or we're always dripping, really slowly. That's a leakage channel. Compare that to those gated channels that we've talked about. The two gated channels that we talked about when we were talking about neurons were those chemically gated channels where I use chemicals to open the gate, and voltage gated channels where they open because of the charge. We also had mechanically gated channels as well, though. When we talk about a mechanically gated channel, how do I open up a mechanically gated channel? What opens these channels up? How do we get these ones open? Yeah, so technically the word that my slide shows is pressure, right? But the way that we talked about mechanically gated channels is we literally push them. The cool thing is now that we've finished Unit 4, now we've actually talked about some examples of where we have mechanically gated channels. What were some of the places in the body that we've now talked about that have mechanically gated channels? Where do I find this kind of channels? Yeah, so Ariel's mentioning they're found on hair cells, not on all of my hair cells, but on hair cells that help me with a couple of my special senses. Yeah, hair cells in the ear. So yeah, it's my hair cells that do hearing and my hair cells that do balance. Yeah, the processes of hearing and balance are the two main places that we see mechanically gated channels being used. So mechanically gated channels, the ones that I push open, now that we've done special senses, we see how that works. I can push them open, or if you remember in the case of when we just finish hearing a sound, I can actually push them closed. So mechanically gated channels, push them all the way open, all the way closed, partially open. That's mechanically gated for you. Voltage gated channels, it's all about the charge. Chemically gated channels, it's all about having that chemical, having that neurotransmitter that we were looking at. I saw a question pop up. Let's see. The question is, in the process of hearing, we only have mechanically potassium channels which depolarize the membrane. So how does the membrane get polarized again? So Pilar, the only ones that we talked about on the cell are those mechanically gated potassium channels. One of the ways that the cells, the hair cells in your ear will repolarize themselves, they will use some leakage channels. I believe they also use voltage-gated sodium channels, ironically. Remember that the fluid that's on the outside of my hair cells makes things funky with the potassium and the sodium stuff. So they're actually flip-flopped on their ions. I believe they also have voltage-gated sodium channels that they use for repolarizing. But the big ones I want you to focus on, we talked about those mechanically gated potassium. There was one other kind of channel that we talked about on hair cells for sure. Does anyone remember the other kind of channel that we mentioned on hair cells? The mechanically gated channels depolarized it. It wasn't actually chemically gated ones. Yeah, we don't have chemicals. Oh, I know what you guys are saying. You're saying with my other senses. Yep, so we do have chemically gated channels. Let's do that really fast. Chemically gated channels. Chemically gated channels found on hair cells who detect what? Hair cells that detect, yep, taste and smell. Yeah, you guys are absolutely right. Detect taste and smell. I was going, yeah, so the words for that absolutely are the odorants, the special type of chemicals, right? That do smell, and then those tastants that do taste, absolutely. So my senses of olfaction, the sense of smell, and gustation, those guys have chemically gated channels. Yep. When we talk about hair cells that do hearing and balance, the other kind of channel that we actually have on those ones, they also have voltage gated channels. What ion do they let inside to help them spit out neurotransmitters? What ion do we bring in to spit out neurotransmitters? Yeah, a couple of us are chiming in. We use voltage-gated calcium channels in the process of hearing and balance as well. If you remember back to that picture of the hair cells, on the hairs, they had the mechanically gated channels, but down a little bit farther on those cells, they also had voltage-gated calcium channels to help them spit out neurotransmitters. So when the charge changed, some of those channels opened up to bring in the calcium. So here's a wink, wink, nudge, nudge. In Unit 1, we defined what these 3 types of channels were. And then as we went through Unit 3 and Unit 4, we used these types of channels to do all kinds of processes in the body. If you can outline for yourself which kinds of channels I use in different things, so with taste and smell, with the process of hearing and balance, with my voltage-gated ones, they're all over the place in neurons. If you can tie together the basic definitions with what I use them for in the body, you're in good shape. That's the goal, is to tie together material from Unit 4 and Unit 3 with this stuff way back here in Unit 1. So use that as you're studying and reminding yourself the difference between the types of gated channels. Where did I use them later in the semester? Where did we talk about voltage-gated channels? Remind myself, how again do I open up a voltage one? How do I open up a chemical one? So if you can tie those things together, that's perfect. That's the goal. Do we have other questions about our types of channels? We talked about the leakage channels and the gated channels. I believe that that's what the learning objective list said to compare those things. You're good. Okay. Let me look at my list here. I guess the one other thing I'll mention too, because we had some questions about transporting things across the membrane. Let me see if I can pull up a picture here, and then we'll talk about bonds. Let's pull up this picture. I'm going to zoom in particular over here on my picture. Remember, we said that this was a super simplified version of the cell membrane. It's never going to look this small, and it's never going to be this perfect like this. But when we were talking about the cell membrane, we said that part of this membrane acts kind of like a bouncer, where it only lets certain things inside the cell. Was that, here, let me draw some lines here. Was that region number 1? I'll put a 1 up top, or region number 2? Who's the bouncer on this membrane that decides what gets to go in or not go in? Yeah, region number 2. It's this middle part down here. Region number 2 decides what can and cannot enter my membrane. When we were talking about what can and cannot enter the membrane, we had a whole list of words that we used to describe things that could get through the membrane, that could pass straight through. What are some of those words that I use to describe things that go straight from outside to inside? Ooh, okay. I am seeing nonpolar and polar. Uh-oh. I am also seeing, I'll put over here, hydrophilic and hydrophobic. So we probably need to remind ourselves what these words mean, because these words are opposites, right? So we need to remind ourselves what these words mean, and then we need to figure out which ones of them can get through. Okay, so let's start with, we'll start with these top 2 up here. Hydrophilic and hydrophobic. Both of these words have to do with how I feel about water, right? That's the hydro part, means water. Hydrophobic, let's start with this one. How do I feel about water if I'm hydrophobic? How do I feel about water? Hydrophobic. Yeah, I don't like it, right? So water hating Water hating, water fearing is technically what phobia is, right? Okay. Hydrophobic, don't like water. Don't play nice with water. I don't want to be by water. Hydrophilic is the opposite of that, right? So hydrophilic means that I'm water loving. I really like to be around water. What helps me to know if something loves water or hates water is whether I would consider it nonpolar or polar. Which of these two words down here means that I love water, that I'm hydrophilic? Is it nonpolar or polar means I love water? Who loves water? Yeah, polar loves water. Polar is the same thing as hydrophilic. Or when we talk about polar, we could say that, again, it plays nice with water. Plays nice with water. Polar things. When I talk about nonpolar, then by process of elimination, that's going to be hydrophobic, meaning I do not play nice with water. Do not play nice with water. When I'm talking about the parts of the membrane and I'm trying to figure out which parts are what, we should remember, we did this in class, where I have water in this picture. Where would I find water when I'm looking at this picture of the cell? Where's the water found in this picture? Do we remember? Okay, yep, I've definitely got water on the outside. So I'll put water. I'll put it over here too, just to remind us that it's everywhere. Water, 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 hooray. And the other place that I do have a lot of water is also on the inside. Yep. The water on the inside is technically making up, remember our fluid, we called it cytosol. So there's a lot of water on the inside and there's a lot of water on the outside, which means that when I look at this membrane, there are some parts that touch water, like part number 1, and some parts that do not touch water, part number 2. That's going to help me to describe or figure out which of these words apply to which places. So here on the outside, I have the parts of, remember that the membrane is made of what are called phospholipids. I have the parts of my phospholipids out here that are called the heads. My heads are touching water. So which of these water words do we think applies to these heads? Which water word applies to the heads, to part number 1? Well, yeah, so we can do the polar word too. Yep. So this part of it would be polar. Yep. This part of it would also be hydrophilic. Hydrophilic, because it's touching water. If I am going to be touching water, if I'm going to be right here next to my water, I better like water because I'm always surrounded by it. If I'm not going to be touching water, which is what's going on with these parts inside here, then I'm totally fine, and actually I will only be hydrophobic. Hydrophobic. I don't like water. So hydrophobic, I don't like water. I'm going to find a place where there's not water. That's where I'm going to live. And remember we said our synonym for hydrophobic was nonpolar. Nonpolar. Nonpolar and hydrophobic mean the same thing. That's these guys here in the middle. The little lipid tails. If you remember, we talked about the heads and the tails. The tails are in the middle. They're nonpolar or they are hydrophobic. They don't play nice with water. My head groups on the outside or on the inside, these ones are hydrophilic. They do play nice with water. And why they can play nice with water is because they are polar. Now, the word polar comes from the types of chemical bonds that they have inside of them. And something that we mentioned we wanted to talk about today were those types of bonds. So let's transition into talking about bonds, but really fast to make sure we get this point. When we talk about stuff that can diffuse straight across the membrane, that can go straight from the outside into the inside, we said that part number 2 was our better, right? We said this is what decides what can and cannot cross the membrane. So remember, when we're thinking about what can and cannot diffuse across the membrane, go straight through the membrane, here's those words: nonpolar and hydrophobic. You've got to match me to be able to pass through. Yeah, so it has to be the same as what's going on here in the middle. You've got to be nonpolar, you've got to be hydrophobic, otherwise you can't sneak through. There was one other word that we used too. The other word that we used to describe it, yeah, Pilar mentioned also you can't be too big. If you're too big, you're not going to be able to fit either. The other word that we use to describe these things is we said things that can pass through are called lipid soluble. Lipid soluble, which is a fancy word for saying I dissolve in fat. Because this is my fatty layer in here, this is my lipids. If you play nice with or you dissolve in lipids, you're good to sneak through as well. So when we're talking about what can and cannot get across the membrane, we'll add a little circle here. It's part of the group. Things that are nonpolar, things that are afraid of water, things that dissolve in lipids, all of those things can get across. Everything else is going to be stuck on the outside in the water or stuck on the inside in the water, unless I use some of those transport proteins that we just looked at. I put protein channels all over this membrane. That's why I said when we looked at this picture, it's never this simple and this easy, because I've got a whole lot of things that are polar and that are hydrophilic and that are water soluble that still have to get in and out of the cell. I have to make special passageways for them to do that because those lipid tails are not going to let it happen. So the things that can diffuse straight through, though, here's our list of descriptions right here. And we've seen these before. Things that are nonpolar, things that are hydrophobic, things that are lipid soluble. Thumbs up or questions? Let me give us a moment to ruminate on this because this is an important idea for us. Got a thumbs up. Questions about the parts of the membrane? Got an A, okay. Awesome, okay. Let's see, I guess that technically was kind of on our list. It was kind of on our list for the day. Okay, well, if you think of questions, you can email them to me or you can bring them to office hours tomorrow as we start to do more of our reviewing. Bring them with you to the session tomorrow. We can talk more about them. One thing you guys asked me to cover were the type of bonds. So let's do some basic review. When we talk about forming chemical bonds, which parts of an atom, over here we've got some atoms here, so here's a hydrogen atom and an oxygen atom. When I talk about making bonds, connecting atoms together, which parts of my atoms am I actually using to build a bond? What do I build bonds with? Chemical bonds, bones. See, I did this yesterday too. Chemical bonds are built with, yeah, a couple of us mentioned it, they're built with electrons. They're built with electrons. When you look at an atom, like my oxygen right here, there are three things that are inside of it. The first one are these electrons that I see floating around here on the outside. These things here on the outside are what I use to build bonds. So we mentioned in class how these electrons aren't just sitting in one place like the picture shows me. They're zipping around in this cloud that I see here on the outside. The color of this cloud tells me how much time they're spending somewhere. So the electrons spend a lot of time by the nucleus here, a little bit less time here in the outside. But I can look at an electron cloud to get an idea of everywhere that they zip around, everywhere that they go. So those electrons are on the outside of an atom. That's what it's going to use to make a bond. In the middle of the atom, in an area that we call the nucleus, we have the opposite of an electron. Does anyone remember the name of the one that's kind of the opposite of the electron? Electrons are negative, so we're looking for that positive one there. Yeah, Pilar's remembering that the opposite of electrons are the protons. Protons. Yep, they have a positive charge. So the protons live in that middle part called the nucleus. What also lives in the nucleus that we didn't talk a lot about, I'm not requiring you to know much about, are what are called the neutrons. And neutrons have no charge. They're neutral. So we'll just put a 0 there. 3 parts to an atom. 2 of them live in the nucleus, the protons and the neutrons. The other ones circle around the outside, called the electrons. So when I form chemical bonds, it's all about those electrons. Electrons in bonds, electrons can be shared, or they can be, well, here, let's put it in polite terms. We'll say transferred. What's the not polite term that I usually use when we're talking about transferring electrons from one atom to another? Do you guys remember? Yeah, I talk about stealing. It makes it stick more for me. I mean, yes, we are technically transferring, but you just stole it from me, man. Like, let's be real. Call it what it is, right? So two general types of bonds we can do. We can do, as we talked about in class, we called them sharing bonds, or we can do stealing bonds. When I talk about the big classification of bonds where we're sharing, does anyone remember the general word for this is a sharing bond? So we got several different answers in here. What I'm looking for with the general word for a sharing bond, as a couple of us have said, is covalent. Covalent bonds are sharing bonds. They're sharing bonds. When we talk about a stealing bond or a transferring bond, those are the ones that I call ionic bonds. These are the stealing bonds. I only have one type of ionic bonds. I just call them ionic bonds. When I talk about covalent bonds, there are 2 different ways that I can share. I can share equally, or I can share not equally. If I'm sharing my electrons equally, the best way I can do it if I'm making a compound, what's that word that I use to describe equal sharing of electrons? Yeah, several of us are chiming in. If I share my electrons equally, we're talking nonpolar covalent. The whole name of the bond is a nonpolar covalent bond. If I share the electrons equally, nonpolar covalent. If I do not share them equally, so not equally, yeah, now I'm going to have those polar covalent bonds. Now, this ties in directly, like we were talking about with the parts of the membrane where we said some parts were polar, some parts were nonpolar. That tells me what kind of chemical bonds they have inside of them. So if I am talking about the head groups on the membrane that we said were polar, that means that the chemical bonds that hold this part together, they're not equal sharing. If I'm talking about that middle part that we said was nonpolar, it means the chemical bonds that hold them together are equally sharing. So when you see those words, polar and nonpolar, used to describe things, that should set off an alarm bell in your mind. Hey, I can predict what kind of bond they have. They have a sharing bond, they have a covalent bond, and either it's going to be equal if it's nonpolar, or it's going to be unequal if it's polar. So here's what an electron looks like on an atom by itself, or an electron cloud looks like on an atom by itself. Remember, we said that places that are darker, the electrons spend a little more time. Places that are lighter, they spend a little less time. I've got a couple of electron cloud images over here. Now notice that we have 2 atoms in both of these clouds. So 2 atoms right here, 2 atoms right here. When I look at these atoms, I can see that their electron clouds are overlapping with each other, right? So normally there would have been just a circle around here by itself, and there would have been a circle around here by itself. But because their circles overlap here in the middle, this is why I've formed a chemical bond. Remember from class, What a chemical bond, the way we described it, is I've got an electron that normally rotates around here that says, oh, I'll rotate around you too. Does a loop over there and then comes back over here. And then maybe it'll go back and do another loop over here. When electrons loop back and forth and don't just go in one circle around one, that's how I build a bond. When you look at these two atoms right here, the size of the clouds that are around them are about the same. It looks like this molecule is equally sharing its electrons. What kind of bond would I have in this molecule since it's equally sharing its electrons? We got some equal sharing going on. Yeah, equal sharing, like I see in this picture here. This is an example of a non-polar covalent bond or a non-polar compound. So non-polar compound. I can tell because my electrons are spending equal time around this one as they are around this one. Because see, when I go down here and look at this particular electron cloud, this cloud is bigger than this one over here. Since this cloud is bigger, my electrons are spending more time here and less time over here, which means that we're not equally sharing. What kind of bond do I see right here? What's the not equal sharing? Not equal sharing, yeah, it's unequal. That means we're looking at a polar covalent bond over here. Polar covalent. So this compound that we're looking at now, we would call this a polar compound. A polar compound. We're not equally sharing our electrons. They spend more time over here than they're spending over here on the other side. Now, we talked about this polar, non-polar stuff in the context of water. When I look at water, the chemical formula for water is H2O. So we've got our oxygen here, and we've got our hydrogens here. When we look at water, does it look like we have a polar or non-polar covalent bond in water? Can we tell from our picture? Polar or non-polar? Yeah, so we're all chiming in correctly that water, H2O, has polar covalent bonds. When I talk about how I figured that out, see how dark my electron cloud is up here, and see how light it is down here by my two hydrogens. What this means is that my oxygen atoms see those electrons a whole lot more regularly than my hydrogen ions do. And this ties in a word that we used when we were talking about bonds here. This is a polar covalent bond. We share things unequally. If you have polar covalent bonds, we get areas that look partially positive and areas that look partially negative. What are those areas called that look partially positive or partially negative? Partially positive or partially negative. Yeah, those are areas that we call dipoles. Dipoles. One of the things that I told you guys in class back in Unit 1 when we were doing this, and this is a reminder, underline, highlight, star. Anytime you've got polar covalent bonds, you will always have dipoles. We will always have places that are partially positive or partially negative. Because the way I form these partial positives or negatives is because I don't share my electrons equally. So if I don't share the electrons equally, which is what polar covalent means, I will generate dipoles. 100%, I will generate dipoles. So any compound like water that has polar covalent bonds is going to be a compound that has dipoles inside of it. Hey, help me out. A compound with non-polar covalent bonds, will this have dipoles too? Do non-polar compounds have dipoles? What do we think? Yeah, nonpolar compounds do not have dipoles. Because if we go down to our definition of what a dipole is, areas where it's partially positive or partially negative, if I share my electrons equally, I don't have anybody who gets the electrons more and is more negative. I don't have anybody who gets them less and is more positive. Nonpolar compounds don't have dipoles because we share the electrons equally. So keep that in mind. Polar covalent, nonpolar covalent, it relates back to those dipoles. The other type of bond that we talked about, I'll toss this out there. Other type of bond that we talked about are these last ones right here. Ionic bonds. What was that word that I used again? When we're talking about an ionic bond, what did we use to describe how these bonds form? Yeah, this is the ones that I like to joke about were the stealing bonds, right? So in a stealing bond, somebody took an electron, somebody transferred, if you will, an electron from one atom to another. Again, I like to call it stealing because it just is more exciting. In an ionic bond, notice how, and I know it's a little bit light, so let me try to, oh, cool. Here, let me make a little circle. I'm learning all kinds of new things about this technology, guys. Check this out. Okay, let's see if I can do this. Oh, that's in the wrong place. So the answer to that question is no. Can I do this? Let's just draw. Okay, right here. Here is one electron cloud. Here is another electron cloud. Notice that unlike up here, where those electron clouds were connected to each other, or up here, where there's just one cloud around this atom or around this molecule, when I talk about these two atoms interacting with each other, their electrons are not shared. We're not sharing. We're sharing up here. We might not be sharing equally, right? Because this one gets those electrons more often. Or up here, we're sharing, and we're sharing them equally because the clouds are the same, but it's one big cloud. When we do an ionic bond, this is not a sharing bond anymore. We should know that ionic bonds are not sharing bonds. So I've got one cloud with a group of electrons that go around this atom, and I've got another cloud where the electrons go around this atom, but they do not go around both of the atoms. They're not going to go around both of these guys. So in an ionic bond, in a stealing bond, this guy gave away an electron to this one over here. Now this one has an extra electron, which means that this one is going to be slightly, or it's going to be completely actually negative. This one has a negative charge. And this one over here has a positive charge because it gave away its electron. Remember when we talked about ionic bonds, we said the basis of these is that opposites attract. My positive is attracted to my negative because my positive literally, it's positive because you see in the picture here, this electron is gone. I gave it away. I now have one less electron than I have protons. That makes me positive. And this one over here now has one more electron than it has protons. That makes it negative. So a big difference between ionic bonds and covalent bonds. Remember, in ionic bonds, we are not sharing electrons. In covalent bonds, we are sharing electrons. Let's see if we remember. Ionic bonds or covalent bonds? Which ones are stronger? Which kind of bond is stronger? Ionic bonds or covalent bonds? Okay, good. Yeah, we are remembering because that's an important idea for us here. Covalent bonds are way, way stronger. Covalent bonds are way stronger. If I'm sharing electrons with you, we're going to stay together. It doesn't matter what you try to dissolve us in. We're going to stay together. We're attached to each other because we're sharing electrons. In an ionic bond, where you took one of my electrons, remember that we said the only thing that really keeps an ionic bond together is the idea that opposites attract. Opposites attract. That's the only thing that keeps this positive attached to this negative. Opposites attracting. So the reason that matters is if I bring in another opposite, which, let's trade pictures here. I bring in another opposite like water that has positive dipoles and negative dipoles. If I bring in something with positive and negative dipoles, and the only thing that kept you, the positive, attached to me, the negative, was the fact that we were opposites, you're going to go and you're going to find some other negatives in water, and you're going to be just as happy because the only thing that kept us together was your opposite charge. And if I'm the negative and you're the positive, and the only thing that kept us together was the fact that you had a positive charge and I was negative, I'm going to find some new positives and I'm going to be just as happy. So when we talk about ionic bonds, we would say that ionic bonds dissolve in water, or they fall apart. They fall apart because the only thing that held these together is you're positive and I'm negative. So let's attach, like that opposites attract. You bring in other opposites, sorry man, we're falling apart. There's no reason for us to stay together anymore. Now, if I'm sharing electrons with you, we can bring in some other positives and negatives. It might change the way that I'm oriented in the water. I might twist around, I might turn my shape a little bit. But if I'm sharing electrons with you, if I'm committed to you, if you will, I like to use the relationship analogies. If I'm committed to you, if we're in a committed relationship, I don't care if there's other positives or negatives around me. We're committed. We're in this together. Whenever we talk about covalent bonds, we'd say do not dissolve in water. It's because they're sharing electrons. If they're sharing electrons, you can bring in other positives and negatives, but they're not going to break my bond. I'm already sharing my electrons. I don't have electrons to spare. It's not just the opposite attracting thing. So make sure we know the difference between how these compounds interact with water. That's one of the learning objectives on our list. Who dissolves? Who doesn't dissolve? Who would play nice with water? Who wouldn't play nice with water? We gave the example, right, of oil and water. When we talk about oil and water, what kind of compound would we say that oil is? Is oil polar or non-polar? When we talk about oil, like vegetable oil, or butter, or those kinds of things. Yeah, when we're talking about oils or fatty substances, those are going to be non-polar. And remind me, since we said it's a non-polar compound, that means that it has non-polar covalent bonds. What's going on with those electrons? If it's a non-polar covalent bond, what's going on with the electrons in those non-polar ones? How do I share my electrons if I'm non-polar? Yeah, one of us is remembering, or a couple of us are remembering, non-polar means we share equally. We share equally. So oil is a compound that I share my electrons equally. We got that cloud that's equal shapes around both of them. If I share my electrons equally, there's no charges. There's no positives and there's no negatives. So you bring in a positive and a negative, like with water. And remember the example I gave you guys in class was, ain't nobody got time for that negativity. I don't need that negativity in my life. Or you bring in, these positives over here. I don't need that kind of positivity in my life. I have a non-polar covalent bond. I'm sharing my electrons equally. Like, back away. I don't want you nearby. So when we think about the way these bonds interact with water, what's going on in water is that water has dipoles. Water has dipoles. Dipoles are these positives and negatives that we see on it. If you have positives and negatives, and if that's the only thing that held you together, like we see here in an ionic bond, then peace out, my bond's gone. If I have dipoles, but I'm sharing my electrons, if there's still something else that's attaching me to a molecule, then I'm not going to fall apart. I'm not going to split up like we see here with the compound. I might play nice with your dipoles. I might get my positives by your negatives and vice versa, but I'm not breaking up for you. You're not worth wrecking my home for, right? And then we talk about the molecules that share their electron equally and they don't have dipoles. You try to put some positivity or negativity next to things that are completely flat and they're completely happy, and they're going to say, I am not going to be by you. Remember the picture that I gave you guys? Draw a little water cup here. When we try to mix oil with water, we end up with all of our water on the bottom. Whoa, that's exciting. Except for the water that's flying away. And we end up with all of our oil on the top. Nonpolar things have no charges. They're sharing their electrons equally. I don't want that positivity or negativity next to me. I'm squishing you down out of the way because I'm perfectly content to live without you. That's when we talk about interactions with water. What questions do we have about these bonds or about the way they interact with water? I know it's a lot to process. It's been a little while since we talked chemistry. Or give me a thumbs up. Do I have any thumbs ups? Or are we still getting there? Okay, well, a few of us have some thumbs up. Some of us are quickly realizing I need to go back and review chemistry tonight, right? I need to get on this chemistry stuff. So if it is confusing for you, because I know I saw a couple of us in the chat that said that this is mixing us up. It's been a long time since we taught chemistry. So go back and look at that. I will also remind you too that if you guys remember, for each of the previous units, there were those videos that I posted for you as supplemental resources. For Unit 4, the supplemental resources are in those guided lessons, which is why you don't have a folder with those videos. But please, for Units 1, 2, and 3, go back to the supplemental resources folders and check those out. I know I've got some good ones about the types of chemical bonds. So if this stuff is tripping you up, please go back and check those out. See if that can help you out. I had a request for the Central Dogma. I also had a request for brown fat, white fat. Why don't I'm gonna hit the Central Dogma right now. I'll hit that today. Do me a favor. I think Emily, was it you that mentioned the two types of fat cells? If that's something that you want to talk about again tomorrow, will you be able to make it tomorrow to talk about those types of fat cells? Because I think once we dive into the Central Dogma, that's probably, okay, perfect. So probably the Central Dogma will eat up the rest of our time today, but I will plan to start with brown fat, white fat tomorrow. So we'll do that topic first tomorrow. I'm going to pull us up the Central Dogma, and we're going to start working on that today. Let's see. First off, let's remind ourselves, see if I have that picture. that the central dogma sounds really scary, but it's not actually as scary as it sounds. So the central dogma, if we were to put it in easy words, let's put it in black up here. So the central dogma, in the most basic terms, is how the cell builds proteins. Let's just summarize it as that. The central dogma is the way that your cells are going to build proteins. So we see down here at the bottom of our picture that a protein can be called a polypeptide, or we can also call it, because when we talk about its little pieces, we can call it an amino acid chain. There's a lot of different ways that we can describe what a protein is. I guess I'll put that word protein under it too, protein. The goal of the central dogma is to help you to take the instructions that your cell has inside the nucleus, called the DNA, and turn those instructions into the particular proteins that you need. So we store our information as DNA, which, if you remember from class, means deoxy, I won't make you type this, ribonucleic acid. That's what DNA is. Deoxyribonucleic acid. I already mentioned this, so let's see how many of us were paying attention. Where does the DNA live in your cells? Where is DNA found in the cells of the body? Uh-oh. I hope we know this one. Okay, well, okay. So Ariel mentioned chromosomes. Yes. I'm meaning big picture, which organelle in the cell. Sorry, I should have said it that way. Yeah, I think that's why we're like, she's asking a really tricky question. No, I apologize. Yes, the DNA of a cell is found inside its nucleus. The DNA has instructions for every single protein that you will ever make in your body. And we want to keep those directions safe. We want to keep them protected. DNA lives inside the nucleus. The problem is, I don't need a lot of proteins in the nucleus. I need most of my proteins out in the part of the cell called the cytoplasm. Now, I'm using that word that I kind of hate because it gets misused, but here's a reminder for us that the cytoplasm is the soupy stuff that we learned. The cytosol in the cell and all the organelles inside of it. The place in my cell where I need to have the proteins that I'm building is outside in the cytoplasm. Some of those proteins go inside organelles and help them do their jobs, like breaking stuff down. Some of those proteins stay inside the cytosol and they give the cell its shape. But all that to say, proteins are needed outside the nucleus. My directions stay inside the nucleus. Yeah, so Eileen mentioned ribosomes. That's a good question. The ribosomes live out here in the cytoplasm. And what's the function of ribosomes? What do ribosomes do for a cell? What's the job of ribosomes in a cell? Yeah, so Rose is mentioning, and it's absolutely right, the most basic job of my ribosomes. Ribosomes are protein builders. Ribosomes are protein builders. So when I talk about the process of the central dogma, I start in the nucleus where I find my DNA. I have to somehow get my directions to those ribosomes that live in the cytoplasm so that they can build me my proteins. The way that I do that is using this molecule that we see here in the middle, mRNA. mRNA, it's named, the M part means messenger. Messenger means I can leave the nucleus. RNA means ribonucleic acid. Messenger RNA, or mRNA, I make it based on what my DNA says. This is my specific directions for the protein that I'm really interested in. I don't want to send out all the directions for every single protein in my body and hope that my cell makes the right one. Because spoiler alert, it wouldn't. It would just make everything and we'd waste a ton of energy. So I make this copy of the specific proteins I want and need in my cell, this mRNA, that comes from my big DNA template. I take these specific directions, turn it into the specific protein that I want. That's the big picture of how the central dogma works. There are two processes I have to do to do this. So taking that genetic information in DNA and turning it into mRNA is a process called transcription. And the way I like to remember transcription is if you've ever seen a doctor who sometimes carries around a little voice recorder, when they're done with a patient, they might stand somewhere and then talk into that recorder. So patient was seen in the office on April 30th. Patient's weight was, 150 pounds. Their blood pressure was 120/80. They go through and talk through the notes. They do still use those things today. Actually, I worked for a doctor not that many years ago, and she would do that, would dictate her note. That information then, that recording, goes to a person, or it goes to a computer sometimes, but a lot of times it's actually people that will listen to that message, listen to that DNA, and turn it into something typed. They transcribe it. So transcribing is taking information that is there in one form and turning it into another form. This process of transcription, we still always stay in what I like to call the language of nucleic acids. So remember in the cell that we had 4 types of macromolecules. 4 macromolecules. One of those macromolecules is what we're talking about right now, called nucleic acids. And these are my info storage. So I'm just going to info. They store information for me. The goal of the central dogma, though, is not to make nucleic acids. I don't just want information. I want proteins, which is the type of macromolecule that I have the most of in the cell. So if I start with information molecules, whether it's my DNA that stores it all, or whether it's my RNA that's a specific transcript of what the DNA said, I still have to take that information molecule and turn it into a protein. That's the process of translation. So think about translation as I'm going to a different language. I'm going from my information storage language to my let's get things done language, and that's the proteins. So the process of the central dogma, going from the information that I store to the protein that I can use, involves transcription and it involves translation. First, I make the RNA, then the RNA into proteins. Here is a wink, wink, nudge, nudge for you, because we took a lot of time in class doing compare and contrast. So we compared things like, where do each of these processes happen in the cell? What organelles are involved in these processes? What kind of molecule, so what macromolecule do I start the process with and end the process with? We took a lot of time in class to do those compare and contrasts. Wink, wink, nudge, nudge, that Dr. Wallace loves to write those kinds of questions, right? So let's do a little bit of that summarizing work together. We'll refer to our notes and see if we can remind ourselves of a few things here. The process of transcription. Where does transcription happen in the cell? Does anyone remember which part of the cell is the location of transcription, where I turn DNA into RNA? Yes, exactly. The process of transcription has to start in the nucleus because that's where the DNA is. So transcription occurs in the nucleus. Translation happens at the organelle that we said helped us to build proteins. Which organelle helps us to build proteins? What was the name of that one? Yeah, Translation happens at the ribosomes. And like we said a little bit earlier, the ribosomes live in the cytoplasm. They live in the cytoplasm. So transcription happens inside the nucleus where the DNA is. Translation happens at those ribosomes because they're the organelles that can actually build proteins. When I start the process of transcription, what does transcription start with? What do I have at the beginning of transcription? What am I using as my template, essentially? Yeah, so Pilar chimed in for me. What I'm using as my template, or how I start transcription, is DNA. I start transcription with DNA. When transcription is done, what have I built at the end of transcription? What did I build? Yeah, so my picture shows me what I built at the end of transcription is that mRNA. I started with my DNA template. I ended with my specific instructions that I need to build a protein. Then I start the process of translation. Translation doesn't start with DNA. What does translation start with? What is the first thing I use in translation? Yeah, now I'm using, I can't do translation without transcription, right? So now I'm using that mRNA that I built to help me build what? What do I end up with at the end of translation? What have I just built at the end of translation? Yeah, I've built a protein. I've built a protein. Eileen mentioned tRNA. tRNA is something that I use to build my proteins. So we should mention them. The other thing that someone mentioned for me was RNA polymerase. So here's the things that we mentioned that didn't make our list so far. tRNA and RNA polymerase. Oops, polymerase. Okay, let's remind ourselves here. What did we say way back at the beginning of office hours was the function of RNA polymerase? What does RNA polymerase do? Well, so it's actually the opposite of breaking down. This polymerase part here has beginning polymer. Polymer actually means I'm building something. Most of my enzymes break stuff down. So I totally get where you guys came up with that from. Most of my enzymes are breakers. When I use this fancy word polymerase on something, that actually means I'm a builder. So RNA polymerase is the enzyme that builds mRNA. It's an enzyme that builds mRNA. So RNA polymerase, would I use that in the process of transcription or the process of translation? RNA polymerase, would I use that in transcription or translation? Yeah, if I have an enzyme that builds mRNA, the step that I built mRNA in was transcription. I use RNA polymerase here, polymerase. Because remember, the goal of this enzyme is to build mRNA. The step that I build mRNA in is transcription. By the time I get to translation, I already have mRNA. I need to turn that mRNA into a protein. So there's no space for me to use RNA polymerase in the process of translation. So RNA polymerase, my enzyme that builds RNA, that's involved in the process of transcription, the first half of the Central Dogma. When we're talking about turning that mRNA, that information molecule, into a protein, we're switching languages here. We have to translate. We're doing translation. tRNA, if you remember, its name technically means transfer RNA. That's technically what it means. But what I told you guys to call it in class was translator RNA. Translator RNA. Because this is a molecule that speaks two languages. It's bilingual. It speaks nucleic acids, like I see right here, and it speaks amino acids, these little pieces that I use to build a protein. tRNA is a process, or is a molecule that I use in translation. I use tRNA here. Because remember, we're calling tRNA my translator RNA. Its job is to take me from information into a usable product. So tRNA is involved in translation. RNA polymerase is involved in transcription. I know I had a question that was about the promoter region. Before I navigate away from this page, I will address promoter region. What other questions do we have about what's written here? Because once I leave this page, it goes away. Yeah, I'll show you, Rosa, the region. It sounds a lot like polymerase, so you're totally cool. Well, here, let me show you guys then. When we talk about the specific directions that we're going to use to build a protein, remember that specific directions are stored inside things called genes. So inside your body, you have genes that code for all kinds of proteins. For the hemoglobin protein that helps your red blood cells, for the actin and myosin proteins that help out your muscle cells. We have genes that give the specific directions for each of those proteins. For us to be able to copy those specific directions, remember that we talked about the importance of the promoter region. And this is a little piece of DNA that lives in front of your specific directions. Remember that we called the promoter region kind of like a billboard or an advertisement, whatever we want to call it. It's a flag that lets RNA polymerase know, hey, if you are really interested in making hemoglobin today, here's where you need to attach, where you need to dock so that you can go through and copy those directions. I would have a different promoter or a different billboard in front of the actin gene. So that promoter region in front of actin would say, hey, if you are interested in building the actin protein, you want to attach here. So each of your genes has a different promoter region to help your body find the particular gene it's looking for so that we only copy the information that we want. Once RNA polymerase finds that promoter region, here's RNA polymerase, the little gray enzyme that I see right here. Once RNA polymerase finds that promoter region and attaches to it, then it's going to go through and it's going to copy all of that information that's here inside the gene. So it copies the information inside the gene, and instead of building DNA like the gene stores it as, it's going to build that RNA molecule like we've been talking about. Here's a reminder for us though. Let me zoom it in. A reminder for us that RNA and DNA are not identical to each other, right? There's a couple of things that we said about RNA and DNA. They're different. Here's the first word that I want to toss out there. Let's see if we can remember. Complementary. Complementary. If I say that RNA polymerase makes a complementary RNA strand to the DNA strand, does that mean that the RNA does or does not completely match the DNA? Are they identical to each other? Yeah, a few of us have chimed in. They are not identical. Complementary, remember the way we talked about it in classes, we said these things are mirror images. They do not match each other, so they are not identical. They're not the same. It's the same way, by the way, when we talk about the fact that your DNA is stored in a double helix, how there's two halves to it. These halves are complementary too. They're not exact matches to each other. So when RNA polymerase takes this DNA message right here and turns it into an RNA message, the way that it's going to do that is by building something complementary. That's a mirror image. So this comes back to our discussion at the beginning of the Central Dogma lecture about which base pairs go together with each other, which ones play nice together. So remember, there's my favorite base pair. Does anyone remember my favorite base pair? Which one's my favorite? My favorite nucleotide. Excuse me, let's be technical. I named my cat after it, if that rings any bells. There we go. Yep. So my favorite base, adenine, my cat. Adenine, A, always pairs with which letter in DNA? Adenine likes to be with thymine. Yep, with T. Adenine and thymine, A and T. My other set of bases are cytosine, that's C, which always likes to chill out with guanine, that's G. When we talk about DNA strands, if I have an A down here, I'm going to put a T up here. If I have a T down here, I'm going to put an A up here. And then I do the same thing with C is always going to be attached to a G, or a G is always going to be attached to a C. That's the way it goes when you build complementary DNA. When I build complementary RNA, which of my base pairs from these four up here, which of these can I not use in RNA? Does anyone remember? Which of these four letters does not play nice with ribose sugar? Yeah, T doesn't play nice with ribose sugar. So every time when I had an A in my DNA, I would end up putting in a U, a uracil. Let's add that over here. In RNA, it's uracil. Is a U. Every time that I have an A in the DNA, now I'm going to put a U in my RNA to because this is the base pair that plays nice with ribose sugar, because that's what makes RNA and DNA different. They have different sugars in them. So thymine is too cool for ribose, but uracil is not. So when I'm talking about complementary RNA to a DNA sequence, you'd build it exactly the same way as you do with DNA, except I can't use thymine. I can only use uracil. Here is a wink, wink, nudge, nudge for you. On the exam, you can expect me to ask you a question where I'm going to make you do just a little bit of typing. I'm going to make you build me complementary DNA or complementary RNA. I'm going to give you half of the double helix. I'll give you a line of letters, and I'm going to ask you to very carefully type in your opposite, your complementary strand. The best way to prepare for those kinds of questions, if you look back at this packet, it says, hey, remember there's a transcription and translation practice activity. That would be a great place. Go through and print that activity again. Make sure you can do that. Or there are lots of practice sequences inside your lecture outline. See if you can do those practice sequences again. I promise it's not hard. We just need to get good at doing it, because like I said, I will have a question or two like that on the exam, just to make sure that we know what complementary means and make sure that we know the difference between complementary DNA versus complementary RNA. We will have to talk about translation more in depth tomorrow. What I do with this RNA strand to turn it into a protein, because we are about out of time for today. So on the agenda for tomorrow, for sure, we'll remind ourselves how translation works. We're going to talk about brown fat and white fat. I do need you guys to do me a favor, and I need you to try to do some studying today of the rest of the stuff in Unit 2, and really start to try to study some stuff from Unit 3, so that we make the best use of our time tomorrow too. So 2 topics for sure tomorrow: translation, brown fat, white fat. And then please bring me some stuff from Unit 3. That's the muscles, that's the neurons, that's the brain. There's all kinds of stuff from that, right? So bring me those things tomorrow, and we will cover those in our office hours then. Are there any last minute questions about our transcription stuff as we wrap up transcription? Or give me a thumbs up if you feel okay. I haven't given you guys a penguin today. Let me draw your little penguin while we're thinking of last minute questions. Getting some thumbs up. We could have our spontaneous dance party if we want to have our dance party for the day. There's our daily penguin. Okay, well, 2 quick reminders, then we'll call it good. Reminder number 1, due tomorrow at 11:59 p.m. Does anyone know what's tomorrow at 11:59 p.m.? Any takers? Friday night. Yep, those articles, current events in anatomy, must be done tomorrow by 11:59 p.m. Again, please plan to take advantage of the fact that you can do extra submissions of it for extra credit. So by tomorrow at 11:59, get those current events in anatomy in for me, those article summaries. Lecture, final, exam opens up Monday morning, can't spell morning, 7:30 a.m. When do we have to have it submitted by? When must it be done? Who knows? Does anyone know when it's due? It's due Tuesday. Yep. Tuesday by midnight. Must be done Tuesday by midnight. And again, remember, we are aiming to try to take it Monday, or at least first thing Tuesday, because you know how your internet can be real tricky on you, right? Especially when it's something like an exam. Your internet likes to go out. So please plan to take it early to give yourself a little bit of a wiggle room. And plus, remember, once you take it, you're done with anatomy. So we want to get it out of the way. So 2 things. By the way, help me out in the chat. What lecture days do I have represented right now? Yeah, till the summer, Eileen says. Yep, I'm in the same boat. Yep, I'm done till the summer, but I'm not actually done because I still have a lot of stuff to do. Okay, I got Monday, Wednesday, got Friday. Do I have any Tuesday, Thursday friends? My Tuesday, Thursday friends don't come to office hours as much. Oh, Pilar's from or no, that's when it's due. Okay, well, do me a favor for my friends in Monday, Wednesday, and Friday. Please, if you could, in your class group me's, if you could just send this out just to remind everyone for me. I really don't want people to miss the current events and anatomy assignment because this is your extra credit opportunity. Oh, awesome. So Pauline, if you're part of group me, send this out to Tuesday, Thursday too. So please make sure everyone gets a text today in the group me. Hey, don't forget current events and anatomy due tomorrow night. I guess the other thing that we should mention too, group wiki, right? Group wiki is due Sunday night. Due Sunday night. So please mention that in the text you're sending out to the group me. If you could just do me a huge favor, because again, I hate it when people miss things. I try to have Blackboard send you out reminders, but it always helps to have as much notice as possible. So send out to your respective group me's for me, current events in anatomy, that group wiki assignment, and when that lecture final is posting, just to make sure that information is out there. All right, well, I am going to shut off the recording for today, and I'll stick around for a couple more minutes here. So goodbye to our friends on the recording. Hope to see you tomorrow for more final exam review.